Sarah.
All right. So good afternoon, everyone. So I'd like to start off by saying that sleep disturbances are amongst the most common comorbidity in neurodevelopmental disorders. And if, oh, pardon. No. Oh, there we go. Okay. And it affects approximately 50 to 90% of diagnosed. And importantly, these disturbances are not simply secondary symptoms. In fact, poor sleep has been shown to amplify many of the core features of neurodevelopmental disorders. Worsening daytime behavioral dysregulation, increasing repetitive behaviors, increasing seizure susceptibility, and reducing the quality of life of both the patients and their family members as well. Yet despite these observations, we still know surprisingly little about whether the underlying circadian system itself. And this question really brings us to, this brings us to the central question that motivated the work that I'm going to share with you all today. And that is, are sleep and circadian disturbances merely a consequence of development, or do they actively contribute to disease pathogenesis? And resolving this distinction is critical, not just for understanding disease mechanisms, but for identifying whether restoring biological timing could represent a meaningful therapeutic strategy. And so in order to understand how timing might affect NDD pathogenesis, I think it's first important to understand that sleep is regulated by two complementary processes. And the first process is the homeostatic sleep drive, or what is referred to as. And this process S is a regulatory mechanism that is based on the prior duration of sleep and wakefulness. Now, on the other hand, we have the circadian system, or process C, and this is largely independent of sleep history, and it represents the action of an endogenous biological pacemaker. And so I think the easiest way to think about this is that the homeostatic system determines our need for sleep, but the circadian system, the optimal timing of that sleep. Pardon? Oh, to that. Okay. Pardon. Now, I would be completely remiss if I didn't emphasize that the circadian system is far more than just a regulator of sleep. In fact, the system represents a hierarchical biological timekeeper that coordinates physiology across scales, from rhythms within individual cells all the way to whole organism behaviors such as the sleep-wake cycle. And by aligning these processes with the Earth's light-dark cycle, the circadian system enables the body predictable environmental changes, thereby optimizing our physiology. And so this coordination begins with specialized retinal ganglion cells that convey photic information to a region of the mammalian brain known as the suprachiasmatic nucleus, or the SCN. And here, the SCN master circadian pacemaker, synchronizing clocks throughout the brain and peripheral tissues of the body to maintain temporal alignment. And so as such, nearly every organ system is under circadian regulation. And when this temporal organization breaks down, the consequences extend just sleep. In fact, circadian disruption has been implicated in various metabolic diseases, cancer, neurodegeneration, and to our lab, neurodevelopmental disorders. And so, of course, a fundamental question is, how is this temporal organization at the cellular level? And so, as I mentioned, virtually every cell in the body contains an autonomous circadian oscillator, built upon an evolutionarily conserved transcriptional translational feedback loop, or what is referred to as the TTFL. And so at the core of the BMAL1 and clock transcription factors that are known to drive or are known to form the positive, which is to say that these two transcription factors heterodimerize and they promote the transcription of thousands of downstream genes, including their own inhibitors, the PER and CRY gene families. And these subsequent PER and CRY proteins dimerize where they translocate back to the nucleus and suppress the transcriptional activity of clock and BMAL1. However, their subsequent ubiquitination and degradation abrogates their inhibitory effect on clock and BMAL1, allowing the cycle to begin anew. And this process takes approximately 24 hours. All right. So how do we begin to dissect how the circadian system or biological timing NDD pathogenesis? And so first, we needed to identify a disorder in which this hypothesis could be rigorously tested. And so we therefore established 4 key criteria. The first is that we wanted to work with a neurodevelopmental disorder that had a strong sleep phenotype, and one in which the sleep was associated with neurodevelopmental trajectory. The third was that we wanted to work with an NDD with an identified monogenetic cause in order to enable mechanistic studies. And then finally, in order to leverage the experimental models we already have in the lab, we wanted to work with a disorder in which the causative gene was ubiquitous and not just restricted to a specific tissue such as the brain, right? And you can imagine that very few neurodevelopmental disorders actually satisfy all 4 of these criteria. However, Kleefstra syndrome fulfilled each and every single one of them, making it the optimal disorder in which to investigate the contribution of the circadian system to neurodevelopmental outcome. And so, of course, I don't need to review any of the clinical features of Kleefstra with anyone in this room, but I would like to report on I would like to focus, on one aspect that immediately caught our attention. In fact, we've kind of spent all morning talking about this one facet, which is that there are several case reports that describe a striking clinical phenomena. And that is that during adolescence, a time of profound reorganization of both sleep and circadian rhythms, some individuals with Kleefstra have been reported to exhibit or experience sleep crises, which are then directly preceded by profound neurodevelopmental regression. And so to us, this raised the possibility that Kleefstra syndrome provides a unique opportunity to understand that relationship between biological timing and neuropsychiatric compromise. And so these observations, led us to establish a collaboration with the BCH Kleefstra Clinic, which, as many of you know, is the largest of its kind in the United States. And here again, I'd like to emphasize that what makes the circadian system uniquely powerful to study is that it can be interrogated across biological scales. And so just as the circadian system organizes biological time from individual cells to whole organism behavior, our experimental approach spans those very same scales, from molecular rhythms in patient-derived cells to rest activity rhythms and clinical individuals with Kleefstra syndrome. And so, as you heard this morning, Dr. Sid's team is leading the clinical phenotyping at the BCH Kleefstra Clinic, and our colleagues in Joe Kozowski's lab are leading the circadian biometrics. And over on the list, we've been involved in profiling the patient-derived samples, but really also in understanding how it regulates the circadian system. And so together, this provides a multi-scale translational framework for understanding how disruptions in biological timing manifest across levels of biological organization. So to start to get into our data, I have to be transparent and say that to date, there are no reported roles for EHMT1 in circadian rhythms or in the regulation of circadian rhythms. But several lines of convergent evidence suggest a connection between the two. The first is that there is a strong bidirectional relationship between histone modifications and circadian rhythmicity. And the second is that there is a publicly available proximity-based interactomic studies that has previously suggested that EHMT1 and one of the core circadian proteins, clock, may be forming protein-protein complexes. And so this leads us to our central hypothesis, which is that disease-relevant mutations in EHMT1 impair circadian rhythms. But we further speculate that the loss of this EHMT1 dependent rhythmicity is a major contributor to that post-pubertal onset of that sleep-associated neurological regression that's seen in Kleefstra. All right, so how do we do this in the lab? So the very first question that we had to ask was, does EHMT1 play a role at all in regulating systems? And so to ask this question, we kind of leverage pipeline we already have in use. And so we primarily work with U2OS cells. They're an osteosarcoma cell line. But what's wonderful about them is that they're beautifully rhythmic. And these cells in our lab contain a circadian reporter, which is the HER2 luciferase reporter. And this enables us to look at circadian rhythms in real time. What we've done here is we've plated these U2OS cells, and the first experiment that we did was try to recapitulate the haploinsufficiency we see in EHMT1. And so we did this by siRNA knockdown, and after a period of incubation, we synchronized the cells and placed them in a luminometer to look at circadian output. And so here what you're seeing are two such traces from what the luminometer or the lumicycle puts out for us. And we're looking at two different graphs over here because we tried two different siRNA constructs targeting distinct regions of the gene. And what was immediately obvious was that we saw a dose-dependent suppression of circadian amplitude, but no changes in the periodicity. Now, this is really, really important because this emphasizes that often the amplitude of the rhythm are thought that it's thought to reflect, a less robust and a less resilient circadian oscillator. So essentially, the system is not as strong. And importantly as well, changes in the amplitude have often been ascribed to changes in that clock BMAL1 portion of the TTFL. And so this was our first indication that EHMT1 might be important for regulating the robustness of the circadian system. And so moving forward, we wanted to corroborate this with another technique. And so what we did here is we utilized a pharmacological inhibitor of EHMT1 known as UNC0642, which I'm sure you are familiar with. And what's great about this drug is that it's highly selective for EHMT1 and its obligate, but it also exhibits low cellular toxicity. And so using a very similar experimental approach, we basically incubated our cells with the UNC0642, placed it back in the lumicycle, and looked at circadian output. And here, what we observed was consistent with our siRNA findings, which is to say we saw suppression of circadian amplitude, but no changes in the periodicity, thereby strengthening our findings that EHMT1 seems to play a role in the strength of the circadian system. All right, so the natural next question that we had was whether EHMT1 itself associated with any of the core circadian proteins. And so we first began to investigate this question by looking at the subcellular localization of EHMT1 and the circadian protein clock. And so here what you're looking at is an immunocytochemistry of both proteins, and then we used high-resolution STED microscopy to look at these cells. And essentially here what you're looking at is EHMT1 in green and clock in magenta, and what you'll observe is that we saw several instances of putative colocalization. And in order to essentially quantify this, we looked at the intensity across individual puncta and found that the intensity profiles overlapped. Again, potentially suggesting that there might be spatial colocalization between both proteins. And I have to be completely transparent and say that while these experiments really provide beautiful images, they don't necessarily directly prove that both proteins are directly interacting with one another. And so to test this question explicitly, we decided to harvest adult mouse brains and immunoprecipitate. So we isolated EHMT1 from the brain, and we asked if clock was found with EHMT1 when we pull it down. Immunoprecipitate. And here what you can see is that if we isolate EHMT1 and we immunoblot for clock, we saw a signal of the correct size, potentially suggesting that these two proteins are forming protein-protein complexes in vivo. But then we also wanted to determine whether this interaction was also occurring in humans. And so we performed the reciprocal experiment in the patient-derived fibroblasts that we had received from the BCH Kleefstra Clinic. And so here what we've done is we've used healthy parental controls. And now what we've done is we've immunoprecipitated for clock, and we find that EHMT1 is also found to interact with clock when we isolate it from these fibroblasts. And so collectively, we've now seen both in vivo and in vitro from the patient-derived samples that EHMT1 and clock seem to be forming protein interactions. So moving forward, now that we had established that EHMT1 was indeed interacting directly with the circadian system, we wanted to know which region of EHMT1 might be important for these interactions. And of course, you can imagine be important later down the line for identifying which patients might be most at risk for losing that interaction between EHMT1 and the circadian system. And so here what I've done is we've kind of taken a blunt hammer approach, and we've divided EHMT1 in 3 main regions in terms of its protein sequence. So as we talked about this morning, there is a large N-terminal intrinsically disordered region, followed by 8 ankyrin repeats, which have been shown to be important for protein-protein interactions. And then finally, there is that C-terminal set domain, which is the catalytic domain of this enzyme. And so I'm going to guide us through now a fairly complicated immunoprecipitation. I'll guide your eyes to the places that are important. But basically, we found that if we expressed full-length EHMT1 and full-length clock, and we immunoprecipitated EHMT1, we found that indeed, again, in this heterologous system, we find that both proteins interact. Great, that recapitulates our in vivo and our in vitro findings. But now what we found was that if we only expressed that N-terminal region of EHMT1, so that's to say we remove the ankyrin repeats, we completely lose this protein's interaction with clock. And so moving forward, what we did now is we performed the same experiment for clock's obligate partner, which is BMAL1. And here what we saw again was that if we expressed full-length EHMT1 and full-length BMAL, that we find that both full-length proteins are indeed interacting. But interestingly, and in converse to what we see with clock, what we found was that if we removed, the intrinsically disordered region of EHMT1, so if we only express the ankyrin repeats and the set domain, now we completely lose interaction with BMAL1. And this is really interesting because it suggests that EHMT1 doesn't engage with through a common interface. Instead, it's different components of the circadian clock through structurally distinct domains, highlighting an unexpected level of specificity in these protein-protein interactions. Oops, did I just end my talk? Sorry. Okay. I was a little premature. All right, so of course, this is an area of future investigation for us, but I think what's really interesting from the is it might hypothesize, and this is something we have to test in the future, that perhaps someone with a mutation in the set domain might be less likely to lose its interaction with than someone who has a mutation in the ankyrin repeats or the IDR. And what's great about this study is that now that we have these biochemical findings, we actually have the rest activity rhythms and the cells from these patients, so we can directly test that hypothesis. All right. So moving forward, I've really focused, with the last few minutes I've had, on what role EHMT1 might be playing in the regulation of the circadian system. But of course, this regulation might be bidirectional, right? And so here, of course, we all know that EHMT1 plays a critical role in development. But beyond that, it's also been well established as a regulator of cellular stress, as well as inflammatory pathways. And this was immediately interesting to us because the circadian system has also been shown to strongly regulate stress pathways and inflammatory pathways. And so what we here is we exposed U2OS cells to either baseline conditions, so just 37 degrees in the incubator, or we exposed them to an oxidative stressor. So we exposed them to sodium arsenite for about an hour. We collect these cells, perform a Western blot, and you can obviously see a twofold increase in EHMT1 in response to that stress. Now, is we out the clock. So we've done this and of course it's right where that's relevant. So we've done this here with BMAL1 knockout cells, but we've also done this with clock knockout cells, and we get the same effect. What you now see is that there's a complete attenuation of this EHMT1 potentiation in response to stress, potentially suggesting that the circadian system, or rather, I think an easier way to say this, that this stress-dependent EHMT1 requires a functional system. And to test this potential relevance to Kleefstra syndrome, we decided to perform the same patient-derived cells that we had. And so what we first did is we started off with the healthy parental controls, again, baseline conditions or sodium arsenite for an hour, and we're able to recapitulate that huge increase of EHMT1 in response. However, now when we do the same experiment in patient cells, we see is attenuated, right? And taken together, again, as I mentioned, our results lead to the high functional circadian oscillator is required for EHMT1 to respond to cellular stress. And these results are really interesting because the patients basically phenocopy what we see when we knock the clock out of cells. And so I'd like to summarize some of the data that required to. Okay. All good? Yeah, go ahead. Perfect. So, as I mentioned, our data suggests that this productive coupling between EHMT1 and the circadian system is required to mount an adaptive stress response. And when this coupling is compromised, cells become less resilient to physiological. And so over time, we hypothesized that this reduced adaptive resilience may contribute to downstream processes like chronic inflammation, metabolic dysfunction, and further disruption of sleep, which is in fact what we see in patients with Kleefstra, ultimately affecting neurodevelopmental trajectory. And of course, I want to be transparent and say that while this model remains hypothetical, it provides both a mechanistic and testable framework linking EHMT1 haploinsufficiency, circadian dysfunction, and adaptive stress responses. And more importantly, it also suggests that circadian dysfunction may not simply be a consequence of Kleefstra syndrome, but an active contributor to disease progression. And so with that, I'd just like to take a quick moment to acknowledge of course, to everyone in the Lipton lab, my supervisor, Jonathan, who is here today, and everyone who is bolded for all of their really hard work in this project, our collaborators, Sid and Joe and their teams, and of course, to our funding sources. And I'd like to, of course, say thank you to the organizers for inviting us to speak today. So thank you.